Good morning. My name is Ariel Louwrier. I'm the CEO and president of a research reagent company called StressMarq Biosciences, and we specialize on making tools for neurodegenerative disorders. And today, my talk is going to be about looking at the effects of sonication on alpha-synuclein preformed fibrils, which are one of the primary resources that we use in order to create some of the models that are required for this type of research. The sonication process itself has been found to be absolutely instrumental in a successful outcome in terms of experimentation. However, the effects of sonication themselves are still a little bit of a mystery. So I'm going to start initially on an introduction to alpha-synuclein fibrils, and look at some of the current best practices, followed by some information on the key aggregation parameters, and then look at some of the information that we've generated in terms of the sonication and, in fact, the solubility of those fibrils, and finally run through some of the conclusions. Now, alpha-synuclein is a small protein. It's about 15 kilodaltons long, 140 amino acids long, and its physiological function is not well understood, but it can act in a prion-like manner, generating a large family of oligomers, which can vary in size and structure, and these can then also go on to form fibrillar constructs, which look a little bit like long strings. The fibrils, as well as oligomers, have been found by a variety of groups around the world to be toxic to cells. And furthermore, the fibrils themselves can be processed, particularly at the C-terminal end, resulting in truncated constructs. Now, in terms of the actual disease, alpha-synuclein oligomers and fibrils can generate Lewy bodies and neurites, which are present in Lewy body dementia, which is a disease often associated with Parkinson's. And these aggregates, which I will show you shortly, manifest themselves in the brain. And they, also synonymous with Parkinson's disease, they kill brain cells. And unfortunately, these aggregates can be transmitted from cell to cell in a variety of different ways. In fact, they're really quite stable entities, causing the disease to spread throughout the brain. Now, the concept of aggregation is not unique to alpha-synuclein. There are others, such as a well-known protein such as A-beta and tau, which can also go through a similar process. And in fact, alpha-synuclein itself, as a proteinaceous seed, can in fact also seed other proteins to go through the same sort of process, including, for instance, gamma-synuclein. The actual disease state of synucleinopathies, as they're known, have no known cure, and that would include Parkinson's, but also diseases such as multiple systems atrophy. Now, on this slide, you will see a schematic taken from Roberts and Brown from a few years ago, and it shows a very useful schematic of how these processes work and how they interact with each other. So we start with monomers in blue on the left, which can then go through a variety of different processes, generating what are called on-path oligomers, which are in red in the middle. And these can vary in size and properties, and then they go on to form these protofibrillar oligomers, which we would probably generally also think, certainly in our laboratories, as soluble filaments, which look very much like the fibrils, but are still in solution, but again have different properties to them, and then finally elongate into what we know as these fibrillar aggregates. There's also the possibilities of having off-path species, and then some of these can be toxic in nature, whilst others can be non-toxic. There's also a real lack of understanding of what happens in the middle of the schematic and how many oligomers and how many different types there are and what their properties might be and how they interact with each other. What is also clear is that there is an element of equilibrium between them. So there is an interchangeability here, which makes the process more complex. The only situation that is clearer is that the fibrils, once they fall out of solution and become a suspension on the far right-hand side, do drive this equation essentially to the right. Now, what does this look like in real life? Well, this is an image from a brain of showing these Lewy bodies and Lewy neurites. The Lewy bodies themselves, initially thought to contain just alpha synuclein, in fact do contain a whole variety of different things, other proteins, but even organelles and membranes. All of these can be stained with a phosphospecific antibody that is specific to alpha synuclein aggregation. This is the Lewy body pathology, which is part of the Lewy body disease, is a devastating neurological disease. How does it work? Well, the substantia nigra, which is part of the brain that controls movement, is a place where alpha synuclein is present. And as you can see at the bottom, these monomers will aggregate into oligomeric forms, and as well as protofibrils and fibrils. And although it's not entirely clear which species has high levels of toxicities, these will affect the dopaminergic neurons and kill them. And as a result, we see the movement disorders that we associate with Parkinson's disease, such as the tremors and so forth. In terms of the protein itself, it's relatively small protein, but has some very interesting aspects to it. So you can see on the left-hand side of this depiction that there's a high degree of alpha helical content, and I draw your attention to the fibrillization core, and that's really an area I refer to as the NAC region, which is really instrumental in the changing of this protein into a different form. And I mentioned earlier that this is prion-like, and what does that mean? And essentially what we're saying is that this has the ability to self-catalyze from one form into another form. And generally we think of it as a benign form into one that is disease-causing. However, that's probably an oversimplification. Regardless, some other interesting aspects of this protein, and we'll go into a little bit more detail in just a moment, is the mutation at position 53. So that's what causes, or one of the causes, certainly, of familial Parkinson's disease. And we find when we make those sorts of fibrils with the mutation at position 53, we find that material to be very aggressive, which is unsurprising under the circumstances. There's also a number of post-translational modifications which are of interest. You can see some of there's 4 tyrosines there that have undergone some study. They are nitrosylated, but probably by far the most interesting one is the serine at position 129. This is phosphorylated in living systems and during the aggregation process. So it's a great way to monitor the evolution of pathology. Generally, if these recombinant materials are used to instigate to the pathology in a given model system, whether it's cells or in vivo or whatever, those materials are not phosphorylated. They're generally made in E. coli. So any pathology that is visualized at a later stage will come from endogenous material. So this is a schematic of, again, taken from the reference cited below, of where we start with alpha synuclein with this alpha helical content, then going through a variety of different changes governed by energy, etc., and ending up with this Betasheet content. And that is a great boon to scientists, because we can measure the evolution of this Betasheet content relatively simply. And if we have a look at this sort of typical graph that looks a little bit like enzymatic reaction with an initial rate here and so forth, we can see the evolution of the blue line as it goes up. And that's the Betasheet that is being created from the alpha helix during this prion-like reaction. And here what we have done is we've taken the seeds of alpha synuclein and we've added more monomer. And these seeds dramatically accelerate the reaction so that there's essentially no lag time. And we can see here that the blue line takes off really rather rapidly. And you can imagine that this is an excellent sort of assay that you might want to screen inhibitors for, because it's a very simple way to see whether or not you would depress that rate or bring it to a halt altogether. Now, without wanting to go into too much detail, I want to emphasize the fact that there are a variety of different types of monomers and oligomers and filaments and fibrils, which can all get a little bit complicated. Often it describes their activities and their structures, but sometimes also in part their surroundings, because it's the surroundings that ultimately dictate what type of structure these will adopt. So we have here at StressMarq, we provide generally two types of monomers, what we refer to as type 1 and type 2, which ultimately will generate type 1 or type 2 fibrils. There's also various types of oligomers. They can be made in several ways, but we generally see them, and we'll look at them shortly, to be roughly 25 nanometers in diameter and of a globular nature. Filaments are what we would think, as most people thinking of protofibrils, but we call them filaments because these are essentially products that are intermediates. So once the oligomeric forms have begun to elongate into filaments, which are these long string-like structures, again, which we'll have a look at in just a moment, they go through a process by which the Betasheet content is not as high as it is with the fibrils. On top of it, they are still soluble. So they do have different properties, although if you do leave them to continue aggregating, they will subsequently form fibrils. And then we have the fibrils themselves. I'm not going to spend too much time on type 3 because it's really, in a way, a hybrid of type 1 and 2. But type 1 and type 2, again, quite different in terms of Betasheet content and also differential properties in biological systems. This is a schematic that gives you an idea of roughly how difficult it is to determine what's what. In the top left-hand corner, we have type 1 fibrils. These would be the ones that are overwhelmingly used in scientific research. And these generate a lot of pathology, for instance, and have high THT fluorescence values. Type 2 is quite different, very little study with this. It does not show a lot of pathology, as we'll see in just a moment. Type 3, I'm not going to worry about too much at this stage. I think the point is it's made differently, and yet under EM, these are of course all electron micrographs, look very, very similar to the others. And then finally the filaments in the bottom right-hand corner, again, looking quite similar to the others. In some instances, particularly on the top left, type 1, and in some of the other micrographs, you can see the association between fibrils, because they are sometimes effectively in duplexes, and in other cases on just single rods. Now, the oligomers look quite different. You may recall earlier I said they resemble these globular structures, roughly circular or so, and it's very easy to discern them from the fibrils themselves, and their properties again are quite different. They don't seed as a general rule. These were induced with dopamine, which is probably the most physiologically relevant chemical that we could induce them with, given the activity of these constructs in the brain. And they don't seed, and yet they have quite a high level of toxicity associated with them. Now, in terms of alpha-synuclein, the first two types of fibrils, if you look at this typical seeding assay, you'll see in purple the very aggressive increase in fluorescence as the type 1 fibril is made. Just below it, you'll see it going at maybe 20–25% of that level of activity is a type 2 fibril. Now, the type 2 fibril and the type 1 fibrils have, in terms of their generation, the exact same parameters being associated with them. So they have the same type of monomer associated with them, the same amounts, the same concentrations, temperatures, etc. But what this clearly shows you is that the type 2 fibril has a much, much lower Betasheet content than the type 1. Now, this is manifest also in the biological sphere. So this is work that has been done in primary rat cells, neocortical cells. And you can see on the left-hand side in the middle, when using a phosphoserine 129 antibody that can detect the phosphorylated endogenous pathology that's been generated in those cells, you'll see when we treat with the type 1, we get a lot of pathology, which is the green picture in the middle. However, if we use type 2, we get essentially nothing. Now, these are relatively short-term experiments, of course. Type 2 has not been particularly well studied for the simple reason that it's generally, at least at this point in time, less interest to the scientific community, principally because the gold standard is seen as that which creates a lot of pathology. But clearly there are different types that do different things. Now, what do these actually look like? Well, these are post-sonication preformed fibrils. On the left-hand side, we have human ones. On the right-hand side, we have mouse ones. Mouse ones generally are seen as being more aggressive in nature when used in cellular or in vivo assays. However, the concept is the same. You'll see that these are roughly in the 30 to 70 nanometer range. They've been declumped quite well. They're quite uniform. And when these are then subsequently injected into rat brains, for instance, as this is a work that was done at a CRO, the alpha-synuclein pathology, again, measured using the phosphoserine 129 antibody, the pathology is generated throughout the brain. And on the top left-hand side, you can see the ipsiperiform. But what's interesting about this is that right below it, you see the contra side. So this is not just something that happens at site of injection. This does spread throughout the injection and very clearly generates the pathology. And this is, of course, of critical importance because for those that are wanting to try treatments or drug regimens to attenuate the process of aggregation and the evolution of this pathology, you have to create the model in order to test those methods or candidates. And I think in this case, the time that is used for this experiment is only 30 days. So it's a very fast way of generating the model, whereas if you were to use transgenic animals, for instance, this could take many months. Now, in terms of the best practices as a good, solid general rule have been very well defined by the Michael J. Fox Foundation, who've been absolutely instrumental and quite extraordinary, in fact, in terms of the input that they have placed from a research perspective and an information dissemination perspective for Parkinson's disease and other associated diseases in this field. the actual handling requirements of the preformed fibrils are indicated very clearly in the reference cited at the bottom, which is only about 18 months old or so at this point. And the key elements that come from that publication are that you need to sonicate fibrils immediately before use, and that it's the active component itself is unclear. However, it's deemed to be short fibrils or oligomers that are made in the process. Now, the size of those, which is a really, really key element of that publication, and others, of course, is that the size must be about 50 nanometers in length or smaller to generate the successful phosphoserine 129 pathology. And if you don't do that, or if you have larger ones, then the success of the experiment is in question, and you will not generate, or the experiment will fail. There is no negative impact, interestingly, of extending the sonication time. So essentially what you're saying is what we're saying is we can't overdo it. And by that same token, there appear to be no differences in amyloid conformation or fibril structure if we sonicate. In other words, the fibril is still the fibril, despite the fact that it is shorter. But it is absolutely key and well understood that these shorter fibrils are more efficient in terms of seeding and their use in vitro and in vivo. Now, the sonication process, being as violent as it is, may negatively affect the stability simply because of the heat generated. So they do request that cooling is required. So if we summarize that briefly, the effects of sonication as we know them is that they reduce the size of the fibrils into smaller segments, which we know to be true. It declumps the fibrils, which we know to be true, and is also probably very intuitive because they are in suspension, they're not in solution, so they do clump together. And we also know, and this is very, very key, that we get a very, very significantly higher activity when we add sonicate or properly sonicated materials, PFF materials, to cells or used in vivo. And the actual difference can be quite extreme. It can really spell the difference between success and failure of the experiment. So our question really was here, is something else occurring? And this really was reflection of the phase status of these experiments. Now, I went back to work that I had done at MIT with Professor Kononov back in the early to mid-90s, where we did something profoundly different, and yet with a few possible parallels to the situation with PFFs. When we use PFFs, they are insoluble aggregates, and yet the substrate that is used to create them is in solution. Now, Alexander Kononov pioneered the use of dried enzyme preparations in non-aqueous media, usually to carry out a variety of hydrolyses or esterifications or trans-esterifications. And by changing different parameters such as the surrounding milieu, he was able to change both enantiomer activities of enzymes or regiospecificities of enzymes and so forth. And so I looked at some of the key criteria that the laboratory and that we used to look at in those days, because we had one key element that we needed to try to answer. And that was, why are those reactions so incredibly slow? You can definitely do interesting things, but the actual speed of reaction can be up to 1 million-fold slower than it is in water. Now, again, very different situation to what we have with fibrils, and yet we know that we dramatically slow down the reaction if we don't sonicate. So I wondered whether there were some parallels here. So I looked at a variety of different criteria that we examined at the time, and I've listed them here. You'll see them all in red and one in white. The ones in red are ones I don't believe pertained, such as diffusional limitations. I mean, we're using water, so there shouldn't be any diffusional limitations through the milieu. We're not really using enzymes, so the active center blockage as well as conformational changes that may result during the drying process aren't relevant here. As well as the transition state destabilization and reduced conformational mobility are not relevant because we're not drying enzymes. Neither is the suboptimal pH situation because, again, we're in water. However, there is this possibility of unfavorable energetics of substrate desolvation because we know that's what happens. We know that a monomeric form of alpha synuclein goes from the fully solvated state to a desolvated state in the fibril. And herein lies our question. If the sonication process doesn't just chop fibrils up or make them shorter, if in fact it has an impact on solubility or solvation of the fibrils, then could the much more successful and higher rates of reaction that we see after we have conducted our sonication process be due not just to shorter fibrils, but to the fact that we may be placing the entire experimental setup into one phase rather than two phases. So rather than having a solvated substrate and a desolvated product, maybe we're putting everything into solution. So we had a look to see if that might be the case, because if indeed everything was in one phase, then the unfavorable energetics of the substrate desolvation would be eliminated because it wouldn't exist, because everything would be in solution. And that increase in prion-like activity that we see would, of course, increase, and that is also known to occur. So does sonication create a single-phase system? Well, we thought we'd have a go, and so we took some of our fibrils, alpha-synuclein fibrils, and this is what they looked like pre-sonication. And the mean size here is just under 70 nanometers, and I'm not going to show you every single step that we did, but just a few of them. But as we increased sonication, we began to see, just as we would expect, the fibrils slowly getting smaller. So this is down to 52 nanometers, and then finally, as we increase time and pulse amplitude, down to just under 40 nanometers. Now, if we look at this in a bar graph format, we can see more or less what we'd expect, sort of a linear relationship as we increase the time and or power of sonication, we decrease the size of the fibrils. That's well known and well understood. The question is, was anything else happening? What was happening to the solubility of these fibrils? So we ran sedimentation assays, and as you can see on the far left-hand side, the two yellow arrows show the amount of material that are in this in the soluble and insoluble phases. And on the far left, you'll see in the supernatant, there's a little bit, but in the pellet, there's the vast overwhelming majority. Now that begins to change as we sonicate. And in our most aggressive sonication protocol on the far right-hand side, you'll see that everything has moved into the soluble phase, into the supernatant, and nothing, indeed, is left in the pellet. Now, what happens over time? Because is this just a transient situation? But if, again, you look at the highlighted areas on the right-hand side, which is representative of our most aggressive sonication method, in yellow, you'll see the difference going from July to October in our pellet, and then looking at our supernatants after storing those materials for 4 months or so at -80, you'll see that the materials stay in solution. So they do not revert. Now that has a number of implications that are contrary to current thinking, because it suggests that you could sonicate and prepare materials and then, for instance, ship them elsewhere, and that you don't actually need to sonicate directly prior to an experiment. It also suggests that whatever we're doing is quite permanent, and that the likelihood of something like that could be something like a covalent modification. Now, we were kind enough to receive some work that was also done by the Andrew West Lab in Alabama in the States, and they were working on sonicating some of our fibrils. And this work sort of complements what we've been looking at. And if we look at the results using dynamic light scattering of sonicating alpha synuclein fibrils directly and quite aggressively, they seem to be able to get them down to a size of about just over 20 nanometers, which is really quite small. Interestingly, if you do the same with gamma synuclein, that's not the case, and further sonication doesn't change this. And it has a much broader distribution and much longer fibrils. Furthermore, beta synuclein has a similar sort of outcome, quite broad, and again, more sonication doesn't actually do anything. Now, these are all quite heavily related to each other with a lot of sequence homology. So again, we thought we'd use a different sonication protocol and just have a look at what was happening from a solubility perspective. This is using a cup horn system, which, at least in our case, is far less efficient than using a probe because we're not in direct contact with the probe head. However, you can see on the left-hand side, we have begun to push some of the material from the control side into that was originally the pellet into the supernatant, just as we would expect. However, beta-synuclein appears to already be in solution, which is something which we had not realized. When making beta-synuclein fibrils, this is a protein that does not fibrilize readily at all. You can try to fibrilize it even for a year without success. What you do need is you really need to hit it with the proverbial sledgehammer and use metal ions and so forth to induce it to fibrilize, which are then, of course, subsequently removed after the fibrils are formed. So they are excellent controls in that regard. But it does seem that they are already in solution and hence don't really benefit from the sonication process because they don't need it. Now, on the final side with gamma synuclein, we see very little effect at all, that the sonication process doesn't really seem to do much of anything. Now, we had a look at the sequences and lined them up, and I just want to highlight the red encapsulation on the sequence at the top. And there we see that's part of the NAC region, which is missing in beta-synuclein, which is why it doesn't fibrilize, because the area is missing. However, it's also quite a hydrophobic area. And so our hypothesis is at this point that the absence of that section is actually sufficient and makes the resulting protein fibrils sufficiently polar that they will actually, naturally, without any further help, go into solution. If we look at a little bit more detail at what happens with gamma synuclein, the major changes are at the C-terminal area. And what I've done is I've highlighted some of the residues in that area, just the methionines and tyrosines, which can undergo oxidative reactions. And if we step back a moment and think about just how aggressive sonication processes are, with the little bubbles that are formed and the high temperatures that are generated when they collapse, you can imagine there's a lot of reactive oxygen species that are generated. And I'm wondering whether just the oxidation of, for instance, the 4 tyrosines in that C-terminal area, which represent to a large degree the changes between these two proteins, if that is enough, if that oxidation is enough to push this into solution, because they'd be able to form more hydrogen bonds and be a more polar construct. Now, one way in which we tried finally to test this was to just treat it at a higher pH. So we took the synuclein fibrils and we also put them into a buffer at pH 10 for an hour on the basis that there are sufficient hydroxyl ions there that if there's something for them to bind, they would probably do that. And if our hypothesis was right, there should be some movement of the insoluble material into soluble material. And interestingly, that's exactly what we see. And that would also, of course, explain the longevity of the sonication process. So we may be looking at not just physical aspects of sonication, but arguably chemical side effects of sonication, which lead to the solubilization of these fibrils. So in conclusion, therefore, the sonication of alpha-synuclein preformed fibrils results in fibrils moving from the insoluble to the soluble phase, and it's likely that the dramatic increase in solubility removes or reduces the limitations that are seen with prion-like activity if that is not carried out. And it's quite possible that the limitations may be due to this unfavorable energetics that are removed by sonication, which are a function of the monomer desolvation into fibrils. There's no doubt that PFFs are in fact reduced in size during sonication. That is definitely a truth, and that the fibril size change may still be a good indicator of whether or not sonication has happened properly. However, probably the best way and the simplest and certainly cheapest way to identify if the fibrils are properly sonicated is to carry out a sedimentation assay and see if in fact they have been dissolved. Also, the fibrils stay in solution for at least 3 or 4 months when stored at -80, and I suspect significantly longer, because it appears that it's likely that we are in fact going through a chemical reaction as well. The sonication process is highly aggressive and does generate the oxidative stress, including the generation of free radicals, and the possibility that we looked at that the free radical generated modification of fibrils plays an instrumental role in the increasing solubility of alpha-synuclein preformed fibrils. That's as far as I'm going to take this. I'd like to acknowledge a few people before I finish. Alexandra Netter-Glangos, the head of our laboratory, was instrumental in the planning of some of these experiments. Sophie Sun was directly involved in carrying out the experiments, and we'd like to thank Andrew West and his laboratory for the data that they provided to us, which was complementary to this work. Thank you very much.